Bismillahirrahmanirrahim. Serotonin R5-HT or 5-hydroxytryptamine. This new series of videos on the autocoids, that is serotonin. Serotonin was named as serotonin because during the clotting of blood, a vasoconstrictor substance was released in the serum, and that vasoconstrictor substance was named as serotonin. Serotonins are also known as antiramine, which was found as smooth muscle stimulant in the intestinal mucosa. In 1951, 5-hydroxytryptamine was identified, and it was confirmed that antiramine and 5-HT are the same metabolite of 5-hydroxytryptophan. This is the amino acid, and L-tryptophan. Serotonin are found in plants, especially banana, pineapple, tomato, animals, and venoms. These substances must be avoided during 5-HT diagnostic test. This is the structure of amino acid L-tryptophan. L-tryptophan is converting into 5-hydroxytryptophan with the help of hydroxylase enzyme. This enzyme inducing one hydroxyl group at position number 5. Look here. And now this is known as 5-hydroxytryptophan. The next enzyme will produce a decarboxylation action, removing this carbon dioxide from here. And this is known as aromatic L-amino acid decarboxylase enzyme. Simply decarboxylase enzyme. This decarboxylase enzyme will remove carboxylic acid, will remove CO2 from here and producing 5-hydroxytryptamine. This is known as 5-HT, 5-hydroxytryptamine. This is hydroxyl group at position number 5. This structure is known as indole, and at position number 3 there is ethylamine. So this 5-HT is also known as indole ethylamine, or it is also known as 5-hydroxytryptamine. This 5-hydroxytryptamine is metabolized through various enzymes. Remember in mind that 5-HT is not absorbed from GIT. This 5-HT cannot cross the blood-brain barrier. However, 5-HT can absorb through special transporter. This 5-HT, 5-hydroxytryptamine, is metabolized through monoamine oxidase enzyme into 5-hydroxyindole acetaldehyde. This 5-hydroxyindole acetaldehyde is then converting into 5-hydroxyindole acetic acid, 5-HIAA, through dehydrogenase enzyme, and this 5-HIAA is then excreting in the urine. A 24-hour collection of urine can be used as indicator for the quantification of level of 5-HT in the blood. Greater this level in urine, greater will be the concentration of 5-HT. This 5-HT is also metabolized to N-acetyl serotonin through aryl amine acetyltransferase, and then acetyl serotonin is converting into melatonin through hydroxy indole O-methyl transferase. 5-HT are mainly 90% found in enterochromaffin like cell. Their synthesis occur, storage occur, and release. And these formed serotonin, they are taken up by platelet through amino transporter. There is no synthesis of serotonin in the platelets. Serotonin is also found in the raphe nuclei of brainstem. This area is supplemented with serotonergic neurons and having a role in sleep, mood, temperature, pain, and appetite control. These serotonergic neurons are also found in enteric nervous system of GIT and around the blood vessel.